A very good evening, everyone. So, myself, Dr. Himani, and today, on behalf of Biotech Agriculture Forum for Technical Education of Farming Society, Kota, Faculty of Veterinary Science and Animal Husbandry, Ranchi, Birsa Agriculture University, Ranchi, I cordially welcome you all to the sixth day of this 7 days international training and workshop program on cutting edge innovations in veterinary science for a sustainable future. So, before starting the today's session, we'll briefly get to know about a little bit about the organizers. So, as we all know that organizers are Vital Biotech. It is India's leading publication, research and training institute whose goal is to tackle the world's most pressing problems which are related to food and agriculture. However, Agriculture Forum for Technical Education of Farming Society Kota, it serves as a spiracle between the network of rural communities in India and other stakeholders such as the government, development organization, and corporate entities. And talking about Birsa Agriculture University, it is a well-known university of India. It was inaugurated in 1981 by then Prime Minister of India, late Srimati Indira Gandhi ji. And primary objective of this university is to develop area-specific technologies and manpower in the field of agriculture, animal husbandry, and forestry for the agricultural development of Jharkhand and economic upliftment of the tribal and other backward classes of the region. The programs and activities of teaching, research, and extensions are being carried out there through 7 faculties and 12 colleges. And there is a College of Biotechnology, 1 center for ABM, 6 directorates, 3 zonal research stations, university farms, 16 Krishi Vigyan Kendras, computer center, central library, and other infrastructure and service units in the university. So the university is well equipped with all the infrastructure, and the College of Veterinary Science and Animal Husbandry, it was established in 1961. And initially it was a part of Ranchi University, and later on it became a part of Rajendra Agriculture University, and then it focused on the UG program only. And later on in 1972, PG courses were added, and it was further expanded in 1977. In 1981, it was incorporated into Birsa Agriculture University and has since offered UG, PG, PhD diploma and certificate courses. So the college is also involved in research related to animal health and nutrition and conducts extension activities like livestock health camps and farmer education. So I would like to take this moment to congratulate the organizers and the entire organizing team for organizing this insightful training program. So today we are highly privileged. We have such honorable dignitaries among us. So dear audience, we are beginning with the session. So our first guest of the today's session is Dr. Abhishek Gupta. So Dr. Abhishek Gupta is an assistant professor at College of Veterinary and Animal Sciences, Rajasthan University of Veterinary and Animal Sciences, Bikaner. Sir holds a MSc in Veterinary Sciences and Animal Husbandry and a PhD in Veterinary Parasitology. Sir has received numerous awards, including the Excellence in Research Award, University Merit Scholarship during PhD at Guru Angad Dev Veterinary and Animal Science University, Ludhiana. Sir has a strong academic and research background, and Dr. Gupta has served as a major advisor and committee member for 17 PG students. His research primarily focuses on parasitic epidemiology, immune diagnosis, and molecular parasitology. Sir has extensive publication record with 62 publications, including research titles, review papers, book chapters, and a book titled Epidemiological and Serological Studies on Tropical Fasciolosis. And the contribution of Dr. Gupta include developing FGCL3 based immunodiagnostics for fasciolosis and conducting studies on uh,
Covid.
cryptosporidiosis and tick-borne diseases. Dr. Gupta is an editorial board member for various journals and served as a reviewer of many reputed journals like Molecular Immunology and International Journal of Livestock Research. Sir has also participated in various major trainings and workshops and presented his research work there. In addition to his research and teaching, Sir is actively involved in community engagement, having organized over 50 training programs and events benefiting thousands of participants. Sir is a life member of multiple professional associations, further demonstrating his commitment to the field of veterinary parasitology. So, Sir, I thankful to you that you have accepted the invitation of organizers and you are here to deliver a talk on such important topic. And so today the participant sir is going to talk on the topic recent developments in diagnosis of hemioprotozoan infection in animals. Sir, with this, I would like to hand over this virtual stage to you so that you can just start your talk and you can just
Thank you, Dr.
inspire our participants with your research work.
Thank you, Dr. Himani. And I would like to ask, I would like to take this opportunity to ask that whether it is important to deliver the lecture in English, or I can use the language whatever they use comfortably.
So, sir,
The
whatever you
important
can use comfortably.
Bilingual can do bilingual.
bilingual. Well, it is up to the listeners because it is important to transfer the message. And if they are more comfortable in
Yes.
bilingual, then I can use them.
You can use, if
Okay.
anybody asks, then you can use different mode.
Oh.
No issue.
I think
Okay.
participants are comfortable with both English and Hindi.
Okay. Okay. So let me start my presentation, and a very good evening to all the participants. And now,
Sir, presentation is not in the slideshow mode.
okay,
Yeah.
let me please. Is it okay now?
Uh, yes, sir. It's in presentation mode, but there is
Blur.
some
It's slightly blur.
yes, I think some text box is added or something like this on the screen.
Yes, that is that window mode I have opted. Should I turn it off?
Yes, sir. Actually, ये पूरा screen पे आ रहा है ऐसा black black.
Okay. Then I'll not be able to visualize you people, but now please let me know whether it is okay now.
Sir, actually the issue is same. Now it's fine.
Is it okay now?
Yes, sir.
Okay. Then
Thank
now
you.
we should start that. Good evening to all the participants, and today I am going to discuss on the topic the recent developments in diagnosis and treatment of hemoprotozoan infections in animals. First of all, I would like to draw your attention to the topic demanding innovations in veterinary science for the sustainable future, as the topic of your workshop is cutting edge innovations in veterinary science for a sustainable future, means that the topic is demanding innovations in veterinary science for a sustainable future. Means what we need is to understand the basic need, its causes, the resources we have, and the best possible solution we can do out of it. Dear participants, we all believe that India has a glory of tackling biggest issues with the available resources, with the power of their innovations and wisdom. So I believe if we could understand the problem, definitely we'll be able to find the way out. So in this PPT, we will try to understand the problem, the approaches we have, the difficulties we are facing in the ground level, and innovations we are looking up to. So first of all, we try to understand what is the hemoprotozoan infections. These are basically the hidden killers in animal blood caused by the protozoan parasites. Caused by the protozoan parasites. Okay. Caused by the protozoan parasites that can infect blood and tissue. They are usually transmitted through arthropod vectors like ticks, mosquitoes, sandflies, testiflies, etc. Affects various animals including cattle, dogs, horses, wildlife, and almost they cover all the livestock, farm animals. And along with this, Rajasthan especially has all the conducive environment to propagate these arthropods vector, and disease transmission is very conducive in this part of the world. Sorry. The common hemoprotozoan parasites which are present in these livestock animals are Babesia, Theileria, Trypanosome, Plasmodium. Uh, Plasmodium is more primarily present in birds and primates, but still it can be counted, and Leishmania. Why it is important? It is important because it not only it causes the visible diseases like anemia, fever, weight loss, organ failure, economic losses, but they several inapparent diseases. They also cause the diseases which are zoonotic, posing serious health risks to humans, and they require early diagnosis, vector control, and effective treatment strategies. One single control strategy or a single approach to stop them is not very effective. Now, it is very important to understand that these hemoprotozoan infections are very near to the rickettsial infections, although they have different uh, origins. They have different, but they have so many similarities in the diagnostics and the pathology. Both are vector-borne, both are transmitted through ticks, mosquitoes, flies, affecting blood and immune cells. Both can cause anemia, fever, lethargy, organ damage, as we have already discussed. The symptoms are common in both the type of diseases. Moreover, both of them are present in the blood. Both of them are very similar. If we are going to diagnose it through microscopy, both of them appears very similar. The approach of diagnosis like microscopy, serology, and molecular diagnostics is very common in case, and both have zoonotic potential and posing serious health risks to animals and humans. So we need to study them together because of if an animal is infected with ticks, it has the chances of getting infected with the hemoprotozoan infections. At the same time, it is also susceptible for the infection or transmission of rickettsial. The co-infections in the environment are very common. The animal which is suffering from Babesia may also be suffering from the anaplasma at the same time because they have common vectors. The symptoms of overlapping means if animal is showing the symptoms like fever, anemia, organ failure, they may be affected from Babesia or they may be affected from anaplasma. This has to be. So mixed diagnosis risk is always there. Holistic One Health approach is essential for the control and prevention. And studying both the groups together improves the diagnosis, treatment, and disease management strategies. And I will like to make a thing clear here that if we are not able to diagnose Babesia and Anaplasma separately, we are not going to decide the treatment part because it is the point where the approach has to be. The true cost of blood parasites, as we have already discussed, they are causing the apparent health illness in animals in the form of anemia, organ failure, or this icterus. But along with this, there are several things which are associated with this, like fever. They are they are causing potentially fatal hyperthermia, which may lead to the death. Severe anemia, after even clearing the parasites from the body, the anemia, if it is not managed properly at time, it may lead to the death. And reducing packed cell volume by up to 70%. If the animal will not die, the productive growth of the animal will be very reduced. Icterus indicating potentially irreversible organ damage and may lead to the death of the animal at a later stage. If we talk about the economic reality, you will have to understand this that the parasite infection in animals two types of problems. One is health wise, which is apparent. The owner can have a look on the health status of the animal that it is reducing and it is deteriorating day by day. At the same time, if the animal is apparently healthy, but the production losses, the production the animal should have given at a point of time may not be achieved due to the subclinical infection going on inside the body. Means the animal is infected but not showing any symptoms. That is called as subclinical infection. Animal is infected and not infected. Sorry, animal is infected and not showing any symptoms. is definitely have a reduced production potential. So the quantified impact is 13.7 billion global annual losses in cattle industry alone due to the tick and tick-borne disease. Treatment costs exceeding more than 300 million US dollars annually in the endemic regions where the hematoprotozoan infections are common. Zoonotic species like Babesia microti cause over 2,000 human cases annually in the United States alone. Now, if we can enlist the hematoprotozoan and rickettsial infections present in animals are Babesia, Theileria, Trypanosome, Plasmodium, Leishmania, Hepatozoon, Cytoxin. In rickettsial, it has Anaplasma, Ehrlichia, Rickettsia, and Coxiella bruneti. All of them are present in India. All of them are having good amount of vectors. The environmental conditions in India are very conducive, and the vectors are responsible for the transmission at almost every state of India, especially Rajasthan, including Rajasthan also. If we talk about the impact of these diseases, the particularly tick-borne infections are major constraint to livestock production and dairy industry in India. These diseases lead to mortality, as I already told you that the apparent effect of these diseases is death, reduced milk yield. If the animal is not showing any apparent health loss, it may lead to the reduced milk yield, which leads to the economic losses to the farmers and decreased animal working capacity. The annual economic losses due to the tick-borne disease in India are estimated up to 498.7 million US dollars. Again, Department of Agricultural Research and Education has estimated a loss of 44,740 million annual losses due to SARA caused by Trypanosoma evansii in India. In India, the annual losses reported due to the tropical theileriosis caused by Theileria is estimated up to 1,295 million US per annum. Anwar in 2018 reported a 57 million US dollars annual losses due to babesiosis and anaplasmosis. One is hematoprotozoal and another is rickettsial, and both of these are responsible for such a huge economic loss in India. Jacob et al. In 2022 told that there is a reduced in there is a reduction in milk in crossbred cows per lactation from 13.91 to 85.34 liters per cow per lactation due to the tick and tick-borne disease in India. So you can assess that along with the health issues, these diseases are very much responsible for the productive losses and economic losses in livestock industry, especially in India. Diagnostic approaches we have. How can we diagnose such diseases in the field level? Now we have four diagnostic approaches in this table. You can see the first column, that is diagnostic method. We are diagnosing these infections by using these four approaches. First is microscopy, where we use the suspected blood and go for the micro staining process. And in the staining process, we study the blood smear and examine the blood smear for the presence of any organism. Second is the serological approach. All of us are well known to this, that these approaches, they use antigens or antibodies for the detection of infections. Another approach to this is molecular approach, where we target the DNA of the organism
So
responsible
actually,
for the infection.
yeah. So actually, slides are appearing little bit like blurry. There is something. I think they are a little bit blurred. There is some
I think uh, that might
now.
be due to the net issues, but
Now
they
it's
are
fine.
clear on
Yeah,
my.
in between it's getting blurred. Okay, you can. Yeah, like right now these are a little bit
Uh,
blurred.
Money, let me know if anything
Yeah,
is
please share the
not
PPT.
going well. Please let. Please let me know, है ना?
Yes, sir.
So should I continue?
Yes, sir.
Okay. So the next approach to this is molecular approach where we target the DNA of the causative organism. And nowadays we are very much aware of use of artificial intelligence in every aspect of our life. So it is at the same time we are also using this advanced imaging and artificial intelligence for the diagnosis of these infections. What we can now use with the best of it is we can use a combination of methods and which can improve accuracy. For example, I'll let you know, for example, if we are using microscopy and there are certain limitations of this step. And after microscopy, if we could if we can confirm this by using molecular approach, that will be of higher accuracy as compared to alone microscope. Now, we will take all these approaches one by one. So first of all, I would like to go with this microscopy. This microscopy, there are several staining methods we use for the detection like Giemsa, Wrights, Leishmans, Fields, and acridine orange stain. Now, I would not take these uh, methods in much detail because they are very common in almost all laboratories. They are using such type of staining methods. All of us are well aware that these Giemsas, Wright, and Leishmans, these are Romanovsky stains, which are a combination of acidic and basic stains, and they highlight the cell structures and parasitic structures so that we can easily differentiate and identify the organisms. The last two, that is field stains and acridine orange fluorescent, these are a bit higher to the basic Romanovsky stains, where we use quick screening and fluorescent. And in this method, we require such higher end equipments for the
Sir,
identification
excuse
of
me,
this.
sir. Sorry to interrupt, but we are still
Mm.
facing the connectivity issue and slides are looking blurry. So I have one suggestion. You
Okay,
can just share the slides to Dr. Dinesh, or he can present through his system. I think there is some problem with the network.
okay. Where
In
I can
between, they
share?
are getting blurry, and so I think
Okay,
participants might not be able to get
there
most
might
of
be.
them.
No, it's okay. Where should I, share? If
Yeah,
they could
I'm
send me a message on WhatsApp or share me a email ID so that I can.
yes. We were just sharing the details in the chat box.
हिमानी इसमें आप दिलवा दीजिए या तो ईमेल आईडी पे व्हाट्सएप पे मेरे दिलवा दीजिए। दिनेश सर अभी ट्रेवलिंग में है।
Okay.
दिस वन आल्सो।
Sir, there is a number in chat box. You can just WhatsApp on that number.
Okay.
Recording in progress.
Sir, have you received that?
Received, sir, received. Just a minute. Sir, is it visible?
Yeah, sir, visible, perfectly visible.
Yes, sir.
Which slide?
Sir, it is 14th slide, sir.
This one, sir. Okay, sir.
14th, sir, 14th.
Okay, this is
No, sir, no. आगे.
Next, sir.
this one.
Yeah,
This
this
one?
one.
Yeah.
Sir, this one?
Yes, sir.
Previous
Okay,
one, I
sir.
think. Yeah, okay.
Yes, sir, yes, sir. So my voice is clear there.
Yes, sir. It's clear.
Sir, just previous slide, sir. Previous to this. Staining methods, sir. Staining methods.
Yes, yes.
Yeah.
Okay. So, I was telling that we are not going to discuss much about these staining methods because they are very commonly used in almost every labs and basic laboratory setups. That Giemsa, Wrights, and Leishman, they are Romanesque stains, the combination of acidic and basic, which are differentiating the cellular structures as well as the organism structures, highlights the organism morphology so that we can easily understand. The last two stains, they are a bit higher end at the higher end because and we need some specialized instruments for the identification uh, that is orange fluorescent type, and we use a fluorescent microscope. uh, if we can if we can apply some modifications like proper smear, if uh, all of us know this, if make by batter smear preparation, definitely accurate results will be showed, and proper fixing and staining time will affect the staining quality. And combination of stains, that is important. Combination of stains can improve the detection efficiency. This has to be explored more. Next to this, after staining, what actually we get these types of slides and infections. If an animal is infected with the infection of trypanosoma evans to such a good extent, then we will get this type of picture. This is in Desquences et al. 2013 who reported this trypanosoma evans, and you can easily visualize the structure in this staining method. But problem arises where the problem arises. That problem arises in the expertise. It is very difficult. It is very important to have an expert eye to identify. For example, if we'll take the example of this infection, that is Ehrlichia. This is the paper reported by Al-Badrani in 2013. You can see this morula stage indicated by this arrow. This morula stage is present in the neutrophil, and neutrophil is a structure which is already having 3 to 5 nucleus, and along with that 3 to 5 lobes, there is a morula stage which is present in the neutrophils, and it has to be identified for the presence of Ehrlichia, and it becomes a bit difficult to diagnose and requires an expert eye. If I'll tell, this is the RBCs and RBCs which are infected by the same parasite, that is Babesia. You can see the different types of images, different types of shapes, different types of stages it is having in the RBCs. Again, this requires a lot of expertise and beautiful staining. If you are lacking that staining standards in your lab,
Dr. Abhishek,
moreover,
once
along with this,
Dr. Abhishek, please let Jitendra know when he has to move the slide, because slides are not moving, and I think you are on next slide.
yes, yes,
Yeah.
yes.
You just
Actually,
send
I
next
have
next
the
one, next one. So I will
Actually,
do that one.
oh,
I will suggest you that you should look on the screen, not on your system, because I think you have opened the slides on your system, and here
the
participants are not getting it properly.
screen you have shared, that is again blurry to me. So
Actually, internet,
I
yeah,
was
yeah,
having
got it. Internet signal is poor that side. That's why. If I can move, then
okay,
it will be, I think after 10 seconds, it will reflect on your side.
okay. Then I'll let you know when it changed so that I'll start
Definitely, sir.
discussing. So,
You can just say
uh,
next. Once you are done with the slide, you can say next, and he will just move on the slides.
Yeah, yeah, yeah.
Yeah,
so
thank
now
you.
on the present, I'll tell you that this is the slide showing the trypanosoma infection in the blood. If you have got good staining facilities and a stable amount of infection in the animal, you will get this type of pictures, and you can easily diagnose the infection in the blood smear. Next, next, please. Now in this slide, you can see this is a 3 to 5 lobular structure, that is a neutrophil, and it is having the morula stage. It is the infection of Ehrlichia reported by Al-Badrani in 2013. You need a very expert eye and a very high quality staining to identify such type of infections. Next slide, please. At the same time, you can visualize this smears. They are infected by the same infection, Babesia microti, reported by Pradeep et al. In 2019. You can see the different shapes of infections, of same infections present in different cells. Again, you require a very good quality staining as well as an expert eye to identify the infection. Next slide. Now, this is the slide showing difference between the hemoprotozoan as well as the rickets cell. Slide A is showing the infection of Theileria. It's a pyroplasmic body infected by the Theileria. And the second, it is showing the infection by Anaplasma. Look, if you don't have any expert, don't have an expertise, that much of expertise, it is very easy for you to misdiagnose the infection. And from here, after the misdiagnosis, the whole treatment and control strategy will be different. So it is very important to have a good quality staining procedures as well as an expert eye. This is the prime limitation of this. We will discuss limitations in detail. Next slide, please. So you witnessed in the previous slides what are the limitations of these microscopy. No doubt it is the golden standard test. If you have diagnosed the infection in the blood smear, then it is 101% sure that the infection is present in the body presently. And there are some limitations which is low sensitivity. So if you think that the animal is infected recently, and the infection has not reached up to the potential that is visible in the infected blood sample, you may misdiagnose the sample as negative. If the parasite load is low, sometimes you may diagnosis as limited. This is the primary limitation of this microscopy. Subjective in interpretation, as we have I showed you in the previous slide, the diagnosis between anaplasma and thylaria. A very skilled personnel is the accurate identification. Differentiation between species can be challenging, leading to the misdiagnosis. Again, the process is very time consuming and requires preparation of blood smear, staining, microscopic examination, and labor intensive. Please, next slide, please. It may lead to false negative, as we have already discussed the infection. If the parasitemia is low or intermittent parasitemia is there, where the infection is not shedded regularly, it may lead to the it may cause undetectable parasites in the single sample. And the sample quality and staining technique may also affect the visibility. There is a limited differentiation ability. Morphologically similar hemoprotozoas, Babesia, the pyroplasmic stages of Babesia and Theileria, they are very difficult. Now, you should remove the trypanosoma from this. I have removed, I think, wrong PPT has been shared. Can be difficult to distinguish under a storm. This is also not suitable for large scale screening. As you have seen, you have to go for the staining of all, and large scanning will show a lot very time consuming process. It cannot also differentiate between a carrier and a subclinical self. If the symptoms are not there and the animals is not having any circulating parasites and still may harbor infection, may lead to the misdiagnosis of the disease. Next. So, keeping in mind these limitations, we have to rush to the next level approach, that is serological. All of us are well known to the serological approach, that these approaches, they are responsible to antigens, circulating antigens and antibodies. So whenever infection occurs in the body, antibodies appear. And we can target antigens for the detection also, but antigens in the body after infection, they are very less in number in the free form. They are always in the bound form. So antibodies are the main components which are present in the free form and can be detected for the presence of infection in the body. So the major ELISA kits or major diagnostics serological diagnosis is based on the antibody detection. The key methods in this is ELISA, as you all know, immunofluorescent antibody test and rapid immunochromatographic tests. Next slide. Uh, if we talk about serological approach, several commercial serological kits are available for the diagnosis of this hemoprotozoan as well as the rickettsial infections in livestock. You can see this cow Babesia ELISA kit. I will not go in details, as these products, they are commercially available. You can go through the internet and can get all the information present in this. Just move on slides to show the products, sir. Next 2-3 slides, they are showing the products only, sir. Next slide, sir, please. This is the cow Theileria antibody ELISA kit. Again, a hemoprotozoan infection of prime importance in India. Commercially available. Next, sir. Again, you can see this is an anaplasma antibody ELISA kit test, complementary ELISA. This is the rickettsial infection, and the commercial antibody ELISA kit is available. With the sensitivity, you can see there's a sensitivity of 100% and specificity of 99.7%. Next step. See, this is important. This is a recent innovation and has been approved by Government of India under the Ministry of Agriculture and Farmer Welfare. You can see that this ELISA used to diagnose Trypanosoma evansi as a multi-recombinant antigen based. Previous to this, we were using this CAT test, card agglutination test for diagnosis of this trypanosoma ivansi, and upgraded the test with this multi-recombinant antigen-based ELISA kit for the diagnosis of trypanosoma ivansi. And these recombinant ELISA kit uses four recombinant proteins like calflagin protein, invariable surface glycoprotein, and tandem repeat protein as well as variable phase glycoprotein. These four recombinant proteins have been used for the diagnosis. Again, this is effective in the diagnosis of trypanosoma ivansi in equines, cattle, buffalo, camel, and dog, etc. And it has a capacity and a shelf life of 6 months and kept at 4 degrees Celsius. So it is a very reliable test, serological test used for the diagnosis of trypanosoma ivansi in animals, approved by Government of India. Next slide, sir. Now, if we discuss about the serology, there are the pros and cons of these approaches. Sensitivity, they can detect low parasitic levels through antibodies, because if the parasitemia is still, we have got an optimum level of antibody and that can be detected. And cons, they may not detect early infections before antibody development. If the antibody titer is not reached up to an optimum level, they will not be tested as positive in ELISA tests. Specificity, ELISA and IFET, they provide species-specific detection. However, if we talk the cons of this, the cross-reactivity is a very important issue in this approach. Speed faster than the molecular methods like PCR and slower than microscopy. If we talk about cost, is a very important sector. More affordable than PCR and making it accessible. Means if we have to spend less to conduct serological testing, the you have to follow the protocol by the company, given by the company, and you will have to report a change in color for the presence of antibodies for the given infection, and cannot differentiate between past and current infections. As you all know that even after the clearance of parasite from the body, antibodies persist for 6 to 9 months or even up to 12 months sometimes. And the test, if the antibodies will remain that up to that optimal level, it will show positive results even after the clearance of parasite from the body. And if we talk about field applicability, serological approaches are very suitable for large scale screening in the endemic areas. And for that also, it requires a specialized reagents, instrumentation, and storage conditions in the field levels. So serology is ideal for screening and epidemiological studies, but PCR and microscopy is needed for the confirmation of active infections. This is the main concern. So we have to rush to another approach, that is molecular. Till studied microscopy and we have gone through the limitations of these approaches. After that, we come up to the serological approaches with the positives. We have also mentioned the limitations of these approaches, and now we have shifted up to the molecular approaches. Please, sir, next slide. Next slide, sir. Okay, sir, we have discussed this slide, sir. Please, next slide. What are molecular approaches? The molecular approach, as we have already discussed, that it can detect the DNA or RNA of hemoprotozoa infection, and they are highly accurate. If you are diagnosing, if you are capturing the DNA of a parasite, that 100% it confirms that the parasite is present, active infection is present, and it is highly accurate. And species identification is also well compared. Key molecular techniques we use for the molecular diagnosis is PCR, as we all know, polymerase chain reaction, quantitative PCR, and group-mediated isothermal amplification. Slide, sir. Why it is better to the earlier approaches means it has high sensitivity and specificity, as we all know that even in low cases it can detect with microscopy mainly. Early detection identifies before the clinical signs appear. Unlike surgical methods, they rely on the as we know that after the infection, the body will get take some time to produce antibodies, and the antibodies will reach to the optimum level after a period of time. Only after that, ELISA can diagnose that infection. But in case of PCR, we can diagnose the PCR, we can diagnose the DNA of that parasite at a very early stage. It can also differentiate its species and strains because it is acting on the because it is interacting DNA at the genetic level, and we can target the species specific primers and can differentiate at the species level. Can reduce the false positive and negatives and more reliable. Why it is better to the earlier approaches. Next slide, sir. Sir, please go ahead, sir. I have to discuss a lot and time is running out. Next slide. Now this is PCR. We all know that it is a really very important and most widely used molecular method for the diagnosis of hemoprotein, and not only hemoprotein. I am talking about hemoprotein. Most of the infection. It is the most important technique that has so widely used in every aspect. It has got three steps. Denaturation. We denature the DNA. After that, annealing, where the primers, the specific primers, they bind to the site, and elongation occurs, which elongates or makes the new copies target DNA. After that, they can be visualized by using this gel electrophoresis. You can see the picture where we're preparing this gel, and in the wells formed, we are loading the products, and they will be visualized using the gel electrophoresis method. Next. Now, if we are so effectively using, sir, next slide, please. If we are very effectively using technique, what are the requirements that are needed for research? Next slide, sir. उनसे पूछ ले उनसे पूछ ले मैम फोन आएगा बाकी सब भर दे मेरे को दे दे चार हाँ लगेंगे 15 10 लगेंगे 10 मिनट लगेंगे So key issues in the PCR implementation is infrastructure challenge. It requires well-equipped labs with thermocyclers and controlled environment. So high financial costs, expensive machines, we require thermocycler, reagents, and their maintenance, which makes it less accessible. Field level limitations. We cannot deploy such instrumentations and huge infrastructure in the field levels. So these facilities remain at high-end labs only, and the ground diagnostic facilities still lacks of this. And actually, we require these facilities at the ground level so that immediate treatment can be used, or effective diagnosis can be done at the field level. Technical expertise need. Complex protocols demand skilled operators for accuracy. And what about scalability concerns? Low throughput compared to rapid diagnostic tests, making mass screening very costly. Each and every cycle is costly, and mass screening cannot be done using PCR, only if it is not very much required. Next slide, sir, please. So keeping these limitations in mind, we are looking up to the next technique, which is a bit higher to this PCR. PCR, that is lamp loop mediated isothermal amplification. Now I'm not going to that much detail, but I'll let you know that how it works, how it is better than PCR. That PCR, you all know, it is having three steps. That is denaturation, which is working on 95 degrees Celsius. Then The next step is annealing, which is occurring at 55, and then elongation, which is acting at 72. Such a sudden changes in the temperature requires high and costly appliances, that is thermocycler. But this lamp technique, this is conducted on a constant temperature ranges from 60 to 65 degrees Celsius, and can be conducted by using simply a water bath or heat blocks. And elongation, this DNA amplification can be done. That is why it is called as loop mediated thermal amplification. It requires a set of 4 to 6 primers comprising of forward and backward inner primers and forward and backward primers and loop primers. They actually bind the target DNA started elongation at the same time and forms a loop, so that the process remains continuous and can be visualized. Also, so this process avoids the use of thermocyclers, and high-end equipments are we can avoid such costly instruments. After that, the products that were visualized by using gel electrophoresis in case of PCR can also be visualized by different methods in this, which are fluorescence method. If we apply dye in the new products, whenever the new product will form, a dye will get darken, and the difference in fluorescence can led to the detection of the lamp products. We can go for calorimetric method, where we can use the optical density difference for the presence of lamp products, or we can use this lateral flow test, where we can develop test and control lines for the detection of lamp products. So these technique is avoiding the use of costly affairs and as well as time. It is also faster than sir. Next slide, please, sir. It is also faster as compared to PCR and requires less infrastructure and can be more useful in field conditions as compared to PCR. In the recent trends, we are shifting from PCR to lamp. But the primer designing is a complex process, as well as the standardization of the lamp at the root level and lab to lab. It is also a very difficult procedure. I would like to tell you or show you some of the studies which are using this calorimetric method, which are using this lamp technique for the detection of thalaria, and the products produced were studied or were captured by this calorimetric method. You can see this paper, that is rapid diagnosis of Theileria annulata by calorimetric loop mediated isothermal amplification assay, and it was proved very useful for the detection of hemoprotozoan infections in field lab. You can note down the details, and you can go through the paper to see the details. And next slide, sir. Next slide, sir. Again, this is a modification to the previous lamp. This is dry lamp, where the use of chemicals have been done to conduct the same test as a dry method, and can be transported to the field levels without any refrigeration, or it can be done at the because the chemicals you have to prepare, and you have to maintain a chain to transport it up to the field level. But by using certain chemicals, you can avoid that cold chain preparation, and that same can be transferred at the room temperature, and the test can be conducted at the ground level. Test can be conducted at the ground level. That technique is called as dry lamp, and it has been effectively used for the diagnosis of bovine tropical theileriosis by Divakar et al. In 2024. It's a recent study, and cross reactivity has also been studied in this method, where it has successfully detected the tropical theileriosis with a high specificity. Next slide, sir. So you can, we have already discussed that. I had to discuss it in detail also that there are several methods, several efficient detection methods of these lamb products, and easily they can be applied at ground level if you are able to standardize the lamb for the diagnosis of these infections. You can also use the detection methods. If we can go to the next slide, please. You can see that such a wide spectrum of antigens it can be so not antigen a wide spectrum of pathogens it can uh, diagnose, and different types of detection methods it can use. It can use this RT-LAMP transcriptase. It can use multiplex where we can diagnose some multiple infections at the same point of time. We can use LAMP on chip. On chip, it can be detected the presence of LAMP products and the positivity and negativity of the samples. We can also use this digital LAMP, and these are the methods we can apply at the field level for the detection of pathogen with the designing of and standardization of LAMP protocols at field level, surpassing the PCR technique, which requires a higher infrastructure. Next slide, sir. Next slide, sir. Yes. If you have understood this LAMP, you might be knowing that it is conducted at a constant temperature of 60 to 65, means the presence of water bath or requirement of water bath or heat blocks. Then another approach to this is recombinant polymerase amplification, which can be conducted at 37 to 32 to 45 degrees Celsius, which means an average of 37 degrees Celsius almost. It can be standardized in temperature level between them. So it's a amplification on room temperature. If you can standardize these techniques, you can amplify the DNA at room temperature. There is no need of this water bath and heat block also. The process is that it has two enzymes primarily. That's recombinase enzyme, which binds to the primers, scans the DNA, fixed to the target DNA, starts amplification with the use of this. When it fixed with the DNA, the single-stranded DNA is separated, and after that, the polymerase start the elongation of the products, and in the similar way, the RPA products they started multiplying they causes the DNA amplification. I'm not going to much details, but you can remember the technique name and you can explore. But here I am insisting to the recent advancements and the basis for which these advancements are to be explored to get these point of care diagnostic test kits, to develop these point of care diagnostic test kits for the use in the field level. So you can use this RPA more effectively than LAMP also. Next up, next slide. Next slide. You can see this development of recombinant polymerase amplification lateral flow for the detection of active trypanosoma vance infection, where it has used the rotat gene 1.2. In the next slide, I am going to show how this RPA has been explored for the detection of this trypanosoma vance in the field. Next slide, sir. Now, see, you can see that this trypanosoma vance is rotat gene 1.2. It has been amplified by using RPA technique, where the primers were conjugated with several indicating, markers like biotin and FITC, FAM probes. These biotin, when the new products will formed, definitely the new product will be having biotin at one end and FAM probe at the other end. And you can one strand will be FAM, will be having FAM probe, and another strand will be having biotin. You can use the detection of these products by capturing the biotin associated with the products. You will capture biotin, and ultimately the product will also be captured, and you can easily visualize in the lateral flow assay. If you can see diagram given below, you will see that sample pad is loaded with the infection, loaded with the susceptible infection, means suspected samples. If it is having the products, it will it will be shown in the test line, because the biotin present in the products will be captured by using antibiotic, that is streptavidin, and it will capture and produce color in the test line. And the similar uh, this FAM that will be captured at control line, and similar lateral flow will let you know about the presence of infection in a very small lateral flow assay, and that can be used by using this technique RPA. And very little expertise is required, as if the color line will develop in the lateral flow assay, the test will be positive, and that can easily be visualized by where the not very technical expertise is not required for the interpretation of positivity or negativity of the infection in this type of assessment. In the next slide, I will try to explain more. Next slide, sir. This is the rapid isothermal duplex real-time recombinase, where you can capture the multiple RPA products at a same lateral flow assay. If you can make two test lines at the same time, targeting two different infections, suspecting in the same sample, you can diagnose the two different types of infections in a same lateral flow assembly or kit you have prepared, and that will be termed as a duplex, or it may be multiplex, where you will have to make three lines, control line, three test lines, and you will have to mark each and every product with different marking base, so that three different test lines will capture three different infections if present that. So that will be termed as multiplex polymerase amplification. And now this paper, this is reporting the duplex recombinase polymerase amplification in equine pyroplasmosis. Next slide, sir. जाना है तो जा, बार बार मत आना। कर दूंगा। This is the paper showing the recombinase polymerase amplification with the basics and applications of these technique in the recent advances. If you can go to the next slide, I will let you know that this technique has led to the shifting of lab from laboratory to suitcase. If you can, sir, next slide, please. Sir, next slide. So see if what this test has given us, what this technique has given us. No, sir, previous, sir, please. I will not take that much of time now. I'll end it up in 5-10 minutes. Previous, sir, previous. Previous to this also. Yes, what this slide have done, you can see that this is the bridge flocculation assay, disposable paper and plastic device, laboratory in a suitcase where we can where we can diagnose these RPA products even in next slide, sir. Next. Yes. You can see this. These are the tests which are not very difficult to diagnose. Various RPA products, they have been combined with several approaches like magnetic beads, colors, dyes, and they are captured at different levels. You can see in the third picture, lab in a suitcase, where it is having this fluorimeter, and they will simply diagnose the fluorescence produced. If the DNA is present, the fluorescence will be altered. Fluorimeter will record the fluorimeter reading and will let you know that the product is positive for the infection or not. Simply in the disposable paper and plastic device, the RPA process will start automatically and will develop a color. In the third multiplex lateral flow assay, you can see we have dropped the sample in the sample pad, and three test lines have been prepared, which are captured separately. And if all the three lines, the color is produced, the infection of the three different infections can be diagnosed in a single assembly. So you can see that these tests have led to the diagnosis to a very extreme level, at a very easier level. If we can do the base work at a lab, we can we can use this test as a point of care. Besides animal, we can diagnose the infection, and the treatment can be started immediately. We need not to send samples to such a high-end labs and wait for the results for a long time. Next, sir. Now, after these diagnostic advancements, we are also going to take up small uh, inventions, small innovations in this medicinal part, treatment part, because we have not much new drugs in the veterinary sector. And the treatment protocols what we follow, that for babesiosis we using diaminogen acetate and imidocarb along with the supportive care. All of us know this. Theileriosis, we use buparvaquinone, which is the drug of choice for the theileriosis, and along with the supportive care. Next slide, sir. Next slide, sir. So for anaplasmosis, we use oxytetracycline and supportive care. Trypanosomiasis, we use diminazine, isometamidine, and supportive care. And these are the protocols we are already following, but we at the same time we are facing the problem resistance to these drugs in the hemoprotozoan infection. So what innovations we can use, we are going to discuss in the next slide, sir. Next slide, sir, please. So we are having much newer choices. So we are using multiple drugs to enhance the therapeutic success. New long-acting buparvaquinone formulations that extend the protection against thyroid, long-acting buparvaquinone. We are also exploring the possibilities of microparticle and nanoparticle drug delivery systems, which are used to improve the bioavailability and prolong drug effects. However, the trials are there, but still the drugs are not very much common in the market. For example, the buparvaquinone loaded nanoparticles, they show higher efficacy, but still in the market we are not getting these types of drugs time. Next, sir. We can also use combination therapies that will be of much used at the present time for babesiosis, or for only one drug, that is diminazine acetate or imidocarb. We can use imidocarb plus oxytetracycline, which improves the parasite clearance and reduces relapse. For anaplasmosis, we can use oxytetracycline along with imidocarb to produce the treatment outcomes in the chronic infections, especially. For trypanosomes, we can use diminazine acetate plus isometamidine for a better long-term control. This is the combinational therapy. If you have learned about, if you have got an idea about synergistic effect, most of them, they are showing the synergistic effect, and they are presenting good results in the infections in the combinational approach. Next, sir. Novel drugs. So triazine derivatives, they are very common in case of human malaria. This triazine, they are emerging as a promising new antiprotozoal, and artemisinin based drugs. Similarly, they are very much effectively used against malaria, especially the resistant malarial cases. And they are also showing a good potential against Babesia and Thalaria infection. The drug repurposing. Most of the drugs, they are used for a specific purpose, but they are also having other qualities. So, for example, levamisole, which is a very good anthelmintic at a dose rate more than 7.5 mg per kg body weight, but at the dose rate less than 2.5 mg per kg body weight, levamisole acts as a very good immuno modulator. At the same time, we can use azithromycin atovaquone, which is presently used in human malaria, shows promising results against Babesia, and Duconate anticoccidial drug is explored for the treatment of Babesia and Thalaria treatment because they all are apoplectins. Next, next slide, sir. Targeted drug delivery system. This is the approach needs to be explored where we can use liposome encapsulated drugs, which can improve the drug uptake by the infected cells while minimizing the side effects. This needs to be explored in the veterinary sector, especially. Similarly, nanomedicine based approaches which can enhance the drug stability, allowing for controlled release and sustainable and prolonged effects. For example, immunocarb loaded liposomes provide a sustained therapeutic effect against Babesia infections in animals. These under exploration, they are not still available for the veterinary prospectus, but trials are there. Trials are being carried out for and further exploration is on. Next. So key challenges, we can understand what the key challenges and what are the gaps. Early differential diagnosis in the infection is very important, and for that we need a combination of approaches as we have already discussed. The only one approach is not. You can use PCR, but for every method you cannot. It's a costly affair. We cannot use. So combination of method is the best approach, and it is very important for us for the to detect the infection at a very early and differential diagnosis. It requires overlapping of clinical signs make it difficult, and it requires advanced molecular methods for accurate detection. Next. So drug resistance, increased resistance to dimetazine, imidocarb, and isometamidine, it reduces the treatment success. And that is why need for alternative therapies like combined drugs and novel molecules, it also requires further exploration. So, if you are discussing the recent advancements in diagnosis, you also need to know about gaps which are to be filled by using these recent by if you are planning a research, you should also know what gaps we should target now. Next. Inaccessible molecular diagnostic. PCR-based diagnostics are expensive and cannot be used widely in the rural areas, and limited field-friendly diagnostic kits are on site. So development of portable, cost-effective, and rapid molecular tests, which we have discussed using LAMP and RPA, they can be standardized for different pathogenic infections present in the areas and make it suitable to develop such tests to be used in the ground level or level. Next, sir. Apps of vaccines making it difficult. Effective vaccines are not available against Babesia, trypanosoma, and anaplasma. Although Thalaria is having a live attenuated vaccine, all of us be knowing that Rakshavak T is available, developed by NDDB and marketed by Indian Immunologicals. But they are also very limitedly available, and research for subunit and recombinant vaccines is still on to provide long-term immunity. So advancements in diagnosis, new drugs, and vaccine research are essential, and integrated approaches combining treatment and vector control and surveillance are needed. Next slide, sir. Future perspectives are portable model diagnostics, as we have, uh, as we have seen in that slides, that use of LAMP and RPA and friendly tests, they are required. They are to be developed and they are to be used for the ground level detection of the infection, and immediate control and treatment therapies should be started. Next. And vaccine development is also another a gap which requires. But if we get some amount of success in developing these portable diagnostic kits, definitely will be having much uh, benefit in this in the developing of these vaccines. The CRISPR-Cas9 editing, it is also very important technique. I might have discussed something about it, but we have already running very late. Next slide, sir. I think I completed most of the conclusive points also. If any question is there from the participants or any queries, they can ask. This RNA interference technique, which is used for the vector control, this is also needs to be discussed a bit, but I think that is enough. I have completed the topic, uh, and I'll I
Thank you,
will
sir. Thank you so much.
welcome any questions or queries from the.
Yes, sir. Thank you for your presentation. And we have indeed got some questions from the participants. So there is a question that please explain hemolymph test of hemioproteasome detection.
Which test?
Hemolymph test of hemioproteasome detection.
Hemolymph test.
Ji, sir.
I'm not getting that infection. Hemolymph means you are trying to visualize the infection in the blood or in the lymph directly. So you can go for staining, you can go for the wet smear, dry smear preparation, microscopy. All options are open. You can directly take the suspected blood. You can bring the blood and take a drop of it on slide, keep a cover slip, and directly visualize if the infection is present in a very good amount. You will see it in the wet smear only. And if you want to make a permanent slide, you can make a dry smear out of it, go for staining procedures, and you can look out for the infection. It will be good for any infection they or they just want to take the to visualize the infection, hemoprotozoite in lymph. Any suspected infection you want to know exactly? Is
Yeah,
it for
sir.
trypanosomes? Is or yeah.
Yes, we have other questions like Dr. Santosh Kumar has asked that in field it's difficult to go through test. What a field veterinarian can do for One Health approach or best to treat the hemioprotozoon and rickettsia, considering a mixed infection.
Yes, that is why I was telling that usually in the field conditions you will get a mixed infection. If you are treating the animal only for a single pathogen, you are not going to get good effects. If we consider a case of a mono infection, suppose you are getting a case of theileria only, and still you are going for buparvaquinone and animal is not showing that good results with buparvaquinone, you will have to shift to the combinational therapy, which we have discussed, that we can use buparvaquinone along with oxytetracycline, or you can use long acting buparvaquinone, as I have already mentioned. But it's a diagnosis that is of much importance. So rather the animal is having theileria only or a mixed infection, because if it is having mixed infection, it will not going to get treated with buparvaquinone only.
Okay, sir. Another question. In your experience, what is the treatment protocol you would suggest for treatment of dirofilaria in dogs?
See, we have got this dirophilia, we have got a drug of choice for dirophilia, na? But the problem is that most of in such case, most of the animals, they don't die of the parasitic infections. They die of the lesions or the effects produced by these infections. So supportive therapy along with the kidney and liver parameter monitoring is very important. if you are getting the infection of dirofilaria, first of all, you will go for diagnosis. If you are confirmed that this is filarial, you can go for diacetaminophen, which is a drug of choice for this filarial worm. Then filarial worm, you can also use DEC, diethyl carbamazine, which can also be used for this. But along with, you will have to monitor the liver and kidney functions of these dogs, because the anemia they are going to produce is severely affecting the liver and kidney. And the antibiotic therapy you are providing along with this will further affect the kidney, and the death will be due to the kidney failure rather than this filarial infection.
Thank you, sir. Thank you for answering the question. We are getting more questions, and I would like to request you that please answer the questions in chat box, as we got too much late for the session. So participants, if you have any question, you can
Yes,
drop
yes,
in
yes.
chat box, and I request sir to answer the questions in chat box.
Sure.
Thank you, sir. Thank you so much. And moving forward, I would like to.
Thank you, sir.
Yeah.
Thank you,
I
sir.
took a lot.
Abhishek, sir. I lost my connection due to mobile discharge. I am out of station, so again thank you for accepting my invitation as a guest speaker. And you nicely explained all those things in a very recent development
No,
whatsoever occurred in your field. Thank you, sir.
it is all mine. Thank you. Thank you so much.
Thank you, everybody. And moving forward, I would like to invite the second guest of today's session, Dr. Brijesh Kumar. Dr. Brijesh Kumar is a senior scientist at Division of Animal Reproduction, ICR, IBRI, Ijatnagar, Bareilly. So, sir has completed his graduation from College of Veterinary and Animal Sciences, Kerala Agriculture University, followed by post-graduation in Animal Reproduction and Gynecology from Pandit Deen Dayal Upadhyay Veterinary and Animal Sciences University, Mathura, and then a PhD from ICR-IVRI in the Division of Animal Reproduction. Before completing his PhD, sir worked as an assistant professor at Pandit Deen Dayal Upadhyay Veterinary and Animal Sciences University, Mathura. Later on, sir was selected as a scientist by ASRB New Delhi and underwent the training for it at ICR Nam, Hyderabad. Then sir joined ICR Research Complex for the Northeastern Hill region, Meghalaya, before being posted at its Sikkim Center, where he served for nearly 5 years and later on joined the ICR-IVRI Ijatnagar, Bareilly. During his tenure in the Northeast, sir worked extensively on high-altitude animal husbandry, focusing on Tibetan sheep and yaks, particularly their characterization in Sikkim and Arunachal Pradesh. Dr. Brajesh has handed over 8 externally funded and 10 institute-funded projects and more than various reputed nationals. It is 1.5 chapters, training manuals, and many more. Sir has developed several gynecological and obstetrical devices with 7 registered designs, including a cyst ablation device and an internal genital injection device and a herd animal catcher. He also holds a patent in collaboration with IIT Kanpur for a cervical softening device in animals with one of his technologies, the cyst ablation device, successfully being commercialized. His contribution extends to the development of a veterinary clinical care app and 2 chatbots, Dairy Shriya and Sheep and Goat Shriya, along with 3 significant educational videos. Recognized for his achievements, sir has been awarded with Young Scientist Award, a senior research fellowship, appreciation letter from Animal Husbandry Departments of Sikkim and UP, and serve as an additional editorial board member of International Journal with extensive field experience. Sir has examined over 4,500 animals in organized farms and field settings, conducted numerous in referral hospitals. His current is focus on embryo transfer techniques, including MOET and IVF-based embryo production, with efforts to enhance blastocyst production rates. He's also involved with the AI unit at the dairy farm, the referral veterinary polyclinic, and the design and development of devices to address reproductive disorders in animals. So your achievements are here.
Okay, ma'am.
Yeah,
Yes,
it's
sir.
fine. So,
Okay, speak before national conference, and I won't take much time, be to 30 minutes, since it's evening time and everybody, especially in Indian continent, everybody is having some other advanced technology for assisted reproductive technology for enhancing animal productivity and in perspective. Slides, is it why it is not moving?
One
so
second.
slides are not
Slides
yet
is not moving. Either
Not
you make
moving.
me host or you have timely go forward.
Excuse me, sir, I didn't get you.
Sir, you are already host, sir.
But I don't know why it is not moving, Dr. Dinesh.
Sir, kindly you share your PPT
Okay,
in
I will
my mail.
reshare. I will reshare.
Okay, okay.
I will want technical reality is the field condition and the potential to be replicated at mass level. Here, some brief about the my all the presentation. If you see that different generation of the ART, if you categorize the artificial insemination and cryopreservation are the first generation ART. If you talk about the second generation after evolution of ultrasonography, synchronization and MOIT program, we put into the second generation of ART, and the third generation ART is the OPU, IVF, and fourth generation ART is the cloning and transgenic animals are there. Cloning and transgenic animal are there. So if you see that if you see talk about the artificial insemination, which is a conventional breeding methods are being used most of the farm and also under field condition. If you will get one calf from one cow almost in a year. If you're using MOIT technology in your for light germplasm, you can get 10 to 12 calves in a year. If you are using OPU, IVF, ET programs under your farm or field condition, you can get 20 to 25 calves in a year from a single donor. So these are the potential of these. You can increase the selection efficiency. You can increase the productivity of your farm in a very short period of time. If you talk about the assisted reproductive techniques, it's just manipulation of spermatozoa or eggs or embryo in laboratory condition to produce viable pregnancy, and primarily used for fertility in the farm animal and to address infertility in the human beings. However, in human being, it can also means it's just vice versa. Here in augmentation of fertility and productivity, we talk in animals and to address the infertility in a human being. In human cases, generally they are going for ICSI, where ageospermia, hypogeospermia are there. There is tubal blockage are there. So many other gynecological issue with females or males. So in that they are using going for the in vitro fertilization and the transfer of the embryo into the female wombs. If you talk about the brief history of the AI in India, the first time AI was done in 1939 by Dr. Sampath Kumaran at Palace Dairy Farm, Mysore, wherein he inseminated a Hallikar cow with semen from the Holstein Friesian bulls and got 33 cow pregnant. So thereafter, Government of India started thought about whether this technology could be used at wide level. In that aspects, they have given one pilot project to IVR in 1942 under the leadership of P. Bhattacharya to test the efficacy of this particular technology, and they found this technology suitable for Indian condition and could be used widely. Thereafter, Government of India established 4 regional semen stations, one in Bangalore, one another in Kolkata, and third one is Patna, and Fourth one is the Montgomery now in Pakistan. So from there now multiple key villages are there. Then slowly chain of AI has been spread all over across the country. If you talk about that bulls, now so many precious bulls, especially dairy belt of our country, Punjab, Haryana, Rajasthan, where farmers are rearing very much prized bull. Even some of our institute like CRB, NDRI, CRC, IBRI, they are also rearing very prized bull. Wherein straw, sometimes if you talk about the CIRB, M29 buffalo bull is there, and farmers are purchasing, means inseminator are purchasing per straw 500, and that too they are supplying only limited to particular person. However, if you see the average price of any straw, frozen semen straw of cattle buffalo in India, 30 to 50 maximally. And seeing that importance of this prized bull, and CIRB have cloned 9 cloned buffalo, and that they are yielding more than 4,600 liters per lactation. Though we are reading our AI coverage is only 30%, but personally I feel it is more than that. Because if you see in and around your locality or in your villages also, everybody is going for the AI only, and more so, except in Buffalo. Buffalo, they are every 3 to 4 villages, they are rearing one Buffalo bull also. Because Buffalo, this frozen semen AI is, consumption is a little less. That's why farmer prefer for Buffalo for natural service. But when awareness is increasing day by day, the importance of AI, advantage of AI, and over the natural service, then definitely slowly this technology also penetrating much and more population of the buffalo also. If you categorize our across the pan India, the AI coverage, so Kerala and Tamil Nadu are the highest with a average coverage more than 75. And our northeastern region, especially hilly states like Arunachal Pradesh, Assam, Manipur, Meghalaya, Sikkim, their AI coverage is less, and more so, they are not very much milk consuming states. They mostly meat, where piggery are more prompt, more integral part of their animal rearing system. So this is the scenario where is our northern India Hindi belts are also in average 20 to 30%. And if you go to the Karnataka, Haryana, and Punjab, they are above the 50%. So altogether, documents say it is only 30% coverage. If you see the recent advances, they have some engineers come up with this camera mounted AI gun. But personally, I feel this is not that much useful. But it could be used as a vaginoscope, and also could be used for training to the new inseminator. Where is the cervix? How the opening, os opening is being seen? Is there any growth is there? Or what kind of the discharge is coming? Whether you can go for AI or not. So these are the things utilized, but it's very specific. You have to use. It's not. It could not be used in a routine manner because it's time taking also, and you may spread the infection through that gun also if you are not making proper sterility and hygienic condition. Other advancement in the semen preservation are cysteine, humine, mitotempo, so many, and low oxygen therapy are a new discovery of these things. But still we are following the conventional for our freezing of our semen, either cattle or buffalo. But these antioxidants are giving very promising results. So hope in future some company or maybe our researchers will come up with some advanced diluent for the semen preservation. If you see the talk about the multiple ovulation and embryo transfer, it is also very much potential and tool for faster multiplication of superior germplasm. Then Heap et al. 1890 transferred story started from Heap, Walter Heap. Actually, he transferred two four-cell Angora rabbit embryo into an inseminated Belgian doe, which gave birth to four Belgian and two Angora young ones. So that sparked, oh, means that could be possible, means uterus are susceptible for other outside embryo also, means they have environment to grow. So from the uh, founding